Hello, everyone. Welcome to this talk. So today I'm going to talk about some research that my colleague and I here at the MRC Cognition and Brain Science Unit are doing about language processing and especially reading. What do we do here at the CBU is to try to understand what parts of the brain are particularly important for reading and also what parts of the brain are particularly important for remembering the information that we read. Knowing this is extremely helpful and informative when we're trying to understand what goes wrong in neurological patients that show deficits related to reading or also memory. So how do we study reading here at the CBU? So there are different techniques that we might want to use to understanding the neural basis of reading, but especially if you're interested in localizing some function, one widely used technique is the functional magnetic resonance imaging technique. So with this technique, we can localize brain function while asking participants to do a task. So they're typically lying in the scanner, like this person here in the picture, and we present them with a task, in this case, a reading task. And we are able, so since we know exactly when we're presenting the task, we are able to measure neural activity while these participants are performing this task. So a second technique that we use here at the CBU is the so-called transcranial magnetic stimulation technique. So this is like a totally, absolutely non-invasive and safe technique that allows us temporarily interfere with the normal neural activity of a specific brain region. So it works basically in this way. So we have the electric current here that travels through a wire into this coil, which has the H-shape figure. And this induces like a strong, powerful magnetic field, which has a particular distribution. If we place this coil and therefore we get this magnetic field close to the scalp, this magnetic field induces an electrical current in the neurons below the scalp and below the center of this coil. So what happens is that, so the way we're using it here at the CBU is that we apply this TMS technique, so this stimulation, to affect temporarily normal functioning on a specific brain region that we think is related to some specific processes, like for instance, reading. And this way we are able to determine whether the role of this area is particularly important for the specific function. And we assess that normally by measuring behavior. So for instance, in this experiment, we use TMS on a specific brain region while participants were reading a task. And then here we were measuring TMS conditions, so condition in which we apply TMS versus no TMS conditions in respect to the amount of time the participants were taking to complete a reading task. So now that I've talked about the different types of techniques that we use here at the CBU to investigate questions about language and reading, I'm going to talk about the specific research that we have recently done here at the CBU. So the main question is, how do we understand what we read? So obviously, likely to understand what we read, we need to access to the meaning of the words that we read. But this is probably not enough. We will probably have to implement cognitive processes that allow us to keep track of this word information over time and to integrate the different words in a sentence. There are also other cognitive processes that we probably likely implement to understand what we read and to remember it. So it turns out the reading is quite complex, and a key and important point here is that our research is trying to distinguish between these different processes, because we think it's important to understand what are the specific origin of reading impairments. So how we did it? So we were interested now first to determine the narrow basis of tracking information over time during reading. So we put our participants in the scanner and we asked them to read some short narratives like this one. So is this narrative? You have to extract the meaning of each word and integrate it with the following word, and you have to do the same at the level of the sentences. So obviously, intuitively, this task requires a bit of tracking of information over time. And in order to know which region in the brain are interested in tracking information over time, we compare this first narrative here with the second narrative, in which participants could not track information over time. In fact, here in this narrative, as you can see, the first part is composed by strings of numbers. So what we did basically was comparing the same narrative part, the same paragraph between a condition which requires the tracking information over time and another condition instead in which integration is not possible with the previous paragraph, so not requiring this sort of cognitive process. And then we looked at the brain activity. So the presentation of these narratives was synced to the recording of brain activity. And our results showed that there is a bilateral network of brain regions that is sensitive to this continuous time extended integration of information over time. So these results are quite in accord with neuropsychological findings, which has shown that patients that have lesions in these brain regions, like for instance, like stroke, aphasic patients, they have linguistic impairments which are related to working memory processes. So those processes actually allow you to track the information over time. So now what about accessing to the meaning of words during narrative reading? So to answer to this question, we use the same technique again, and now we have asked to our participants to read the same narrative that we presented in the previous slide. However, this time this narrative was compared to another narrative. And the crucial difference between these two narratives was not related to the amount of information that participants had to track over time, but rather the content. So this first narrative was composed of two paragraphs that were coherent in terms of meaning, whereas instead the second narrative was presenting two paragraphs which were quite inconsistent in terms of semantic meaning. So we compared these two conditions and we looked at which brain regions was sensitive to semantic manipulation. So our results I'm presenting to you here in the bottom part of this slide are those relative to the brain region that respond to this semantic manipulation. So here we had a network of brain regions, which is again quite bilateral, but there are different brain regions involved as compared to the brain regions that are supporting instead the tracking of information over time. Indeed, since the semantic processing recruits only the activity of this part of the brain, which is the anterior temporal lobe, and this result is quite consistent with what we know from neurological patients, especially patients that have semantic dementia, who are experiencing this semantic impairments. So for instance, they are not able to retrieve the meaning of the words or the meaning of the picture. so they can't recognize objects, and this seems to be due to brain damage in this brain region. So to sum up what we did find, so we found that the inferior frontal lobe and the anterior temporal lobe are supporting different type of cognitive processes for reading, and particularly tracking of information over time and accessing to the meaning of words, respectively. What is still not sure and undecided is the role of the parietal cortex during language processing. And this question mark has basically motivated some further research that we did here at the CBU. And particularly, we investigated the possibility that the inferior parietal cortex is actually important for encoding information during reading. So in order to investigate this question, this time we used the transcranial magnetic stimulation technique, so the abbreviated for TMS. And TMS was applied on the scalp of our participants to interfere with the normal functioning of the inferior parietal cortex while they were reading the same narratives that we used in the previous experiment in which we used fMRI. And the idea here was to see whether applying TMS on the parietal cortex was affecting the ability of participants to retrieve some information about the content of these narratives. And particularly, we measured how long they were taking to respond to these memory questions. So what we found is that when you apply TMS on the parietal cortex, as compared to when you TMS during the same reading task is applied to a control area, participants are taking longer in order to respond to the questions about the content of the narratives. So having said that, the research that so far I presented to you is showing that reading is a complex process, which involves different type of cognitive functions, including functions that allow you to access to the meaning of words and the sentences, working memory functions, which allow you to track the ongoing information that evolves over time, as well as mechanisms and processes that allow you to encode the content of the narratives or any type of verbal material you're presented with. So having said this, I would like to conclude this talk by thanking all of you for your attention, and I wish you a great day.